All right, good afternoon, morning, or evening, depending on where you all are zooming in from today. My name is Dana Delanoy, and it's my distinct pleasure to welcome all of you today to this virtual symposium on per and polyfluoroalkyl substances, or PFAS. This symposium is sponsored by the Michigan Center on Life Stage Environmental Exposures and Disease, or what we like to call M-LEEaD for short. And this center is a National Institute of Environmental Health Sciences core center. The network of core centers brings scientists together who seek to improve our understanding of the contribution of exposures towards the etiology of chronic diseases like asthma, neurodegenerative disease, and others. Today's virtual symposium focuses on issues related to the exposure, toxicity, and policy in the unfolding PFAS contamination across Michigan and globally. And it builds upon a successful day-long researchers and community partners symposium M-LEEaD held in collaboration with the Emory Hercules Center back in February of 2020, just before the COVID pandemic took hold and changed our way of gathering and way of communicating science. I'd like to thank our M-LEEaD Center leadership, and in particular, Vasantha Padmanabhan and Jacqueline Goodrich for organizing today's symposium. I'd also like to thank Michelle Dowd, Robin Wiley, and Meredith McGee for all of the logistical support. And finally, I'd like to thank our speakers for sharing their expertise and time with us today, and to thank you, our virtual audience, for your participation and interest in this important topic. Our symposium today will feature 3 speakers who will each focus on one of the 3 topics: exposure, toxicity, and policy. To kick off the event, I'd like to introduce Dr. Jacqueline Goodrich, a research assistant professor of environmental health sciences. Dr. Goodrich's research interests combine toxicology, epidemiology, and epigenomics with the goal of identifying epigenetically labile regions that contribute to environmentally induced disease susceptibility at various life stages. She has been working on issues related to PFAS in both childhood and occupational cohorts. Jackie, I'll turn the virtual Zoom podium over to you. Thank you all.
Thank you, Dr. Delanoy, and thank you, Emily, for sponsoring this event. Before we get going, I just want to give the audience a couple of Zoom housekeeping tips. We're in webinar mode, so you won't be able to use your microphone or your video. However, if you do have questions, you should have a Q&A option that you can see it at the bottom right there. And at any time during the symposium, put your questions in there and we'll scroll through them and try to get as many of them answered as possible at the very end in our question and answer session. If the meeting is disconnected, stay in, it should automatically reconnect. If anybody needs closed captioning, please put a note to me in the question and answer box and we'll make sure to get that turned off. We're also going to put a recorded version of this webinar on our M-LEEaD website, and that version will have closed captioning to go with it too. And it'll be my pleasure to hear presentations from our speakers in a few minutes, but I want to give a very quick overview of PFAS for just a couple of minutes before we do that, and also thank my co-host Vasantha Padmanabhan for help in getting this idea together today. Now, for those of you who are here, you probably know why we're concerned about PFAS, and that's why you're here today, but just in case, I want to give a very short background on it. Why do we care about PFAS? They're absolutely everywhere. So there are many of these chemicals. Many of them are known as forever chemicals because they can be quite hard to break down, some of them nearly impossible to break down, and so they persist in the environment for a long time. The first few PFAS ever made were made as early as the 1940s, and they were thought in some ways to be miracle chemicals because they have really neat properties. Some of them are flame retardant, they can be resistant to stains or grease and other things. They have really great properties for use in industry. However, they can be quite toxic to human health and hard to get rid of. And because of that, we find them in water sources, in fish and seafood, and then of course in the industrial and commercial products they're used for on purpose, like rainproof gear, Teflon pans, food packaging, and things like that. So how many of these are there? There are different estimates of this, but there's thought to be at least 4,000 different PFAS that have ever been made. And as late as 2019, the EPA estimated that About 600 of them are used currently somehow in the US. But what about toxicity? Research on PFAS have really only been going on, or have only really been public knowledge the last few decades, and most of it is on two, PFOS and PFOA, which are 8 carbon length PFAS. And we do know that these two are toxic, but there's not much known about many of these thousands of others. And so, what do we know from those couple that have been studied? We know that there's just a myriad of health effects associated with these, from effects on developing children if they're exposed in utero, to effects in women and men, including thyroid diseases, dyslipidemia, cancers, liver damage, kidney damage, and some reproductive effects as well. And something that you'll get to hear about today is the immunotoxicity that has been found for several PFAS. So where do we go from here? There are a ton of things to do in, as far as PFAS science and policy goes, and many of you attending this webinar today are working on some of these things. Of course, of utmost importance is protection of exposed communities and cleanup of exposed water sources and environments. We need toxicity characterization of the thousands of PFAS that we know nothing about, and risk assessment for these as well. And we need regulations to protect populations around the world from these levels that are just everywhere. And of course, ultimately, industry needs to develop some kind of safe alternatives that aren't just other PFAS in the future. And there's so much to do, but really today, with only a 2-hour webinar, we'll only focus on a couple of things in this broad field. Our first speaker will talk about the burden of PFAS exposure around the world. Our second speaker will talk about immune toxicity, and the third speaker will give some examples of what's going on in the policy realm and the community realm. And then we'll get to have a question and answer session where we hope to have a great discussion on all of the questions our audience has about PFAS. And so with that, I would like to introduce our first speaker. Dr. Elsie Sunderland is a professor of environmental science and engineering in the Department of Environmental Health at Harvard. She's also the Gordon McKay Professor of Environmental Chemistry. She studies how biogeochemical processes affect the fate, transport, and bioaccumulation of contaminants in aquatic ecosystems. She's worked on mercury and other toxicants and is now doing a lot of work on PFAS as well. She did her PhD in environmental toxicology and then went on to work at the US EPA on air pollution policy and regulation before joining the faculty at Harvard. There, she's involved with the Center for Environment and the Harvard Center for Risk Analysis. Her research group develops and applies models to characterize how changes in climate or environmental contaminants affect human and ecological health. And today she'll be talking about the global burden of exposures to PFAS.
Sorry, a little trouble finding the unmute button in full screen mode. So I hope you can all see these slides, and thank you so much, Jackie, for the introduction and for the organizers, to the organizers for the invitation to be here today. So I'm going to talk a little bit about what do we understand currently about exposure sources to PFAS, and Jackie just provided a nice little lead in with the diversity of those sources. And then I'll end talking a little bit about, what is kind of the global picture for these compounds. And what I'm showing you in this slide is a picture of the Faroe Islands. I don't know how many of you have had the pleasure to travel there, but it's a beautiful place, and it's also a site where we've been able to look in detail at some of these exposure sources, and some of my colleagues have studied now the health outcomes associated with PFAS in the same location. So when we think about PFAS, they're very interesting as a toxicant class because we know that they have local exposure pathways associated with large industries that are impacting our communities across the country. And they also have a global pathway. So we see them at high levels in some cases. In biota in the Arctic, in the Southern Ocean, we can detect them. So they're an interesting contrast in spatial scales. And I'll talk a little bit about this today. I'd like to start by just thinking about the actual pathways, the human exposure pathways to PFAS. And as Jackie mentioned, these are quite diverse. This is a cartoon we put together for a review paper almost 2 years ago now. Time passes very quickly. And the idea here that I want to get you all thinking about is that, we have these large point sources, and we have manufacturing locations that have contaminated the environment, but the pathways take two different sort of courses to reach us as humans when we think about exposure. And I want to separate your thinking that way, because when the PFAS that we produce industrially and enter something like a consumer product, we're exposed to that, of course. But if we decided we didn't want it in that product anymore, as we're seeing in Europe right now, we can say, hey, these aren't the best compounds for our health. We'll phase them out, and we can expect to see a very rapid response or change in exposure versus some of these environmental pathways. So one of the main ones has been the use of PFAS in firefighting foams, because they're so effective at fighting certain types of fires. And these sort of sit in the environment, travel in the groundwater for long periods of time, through rivers, eventually reach the ocean, which is thought to be the terminal sink for most PFAS that we release through human activity, and then gradually work their way into our drinking water or our food through bioaccumulation in seafood and other things. So these are different time scales of processes that we have to think about. So right now we have two different things that we need to think about when we talk about PFAS exposure and how to mitigate risks associated with PFAS exposure. The first is really thinking about what are the products and ongoing new exposures to PFAS, and the second is how do we deal with the legacy PFAS contamination that we have in the environment. We do that by thinking about these different individual sources that I've outlined here. The ones that my group has thought about in some detail include drinking water, seafood, and consumer products, and I'll touch on this briefly now. But there are many, and so others needing attention that we have very poor understanding of or data for now include many different dietary sources through other foods. Dust in the indoor environment has been studied a little, but I believe we need much more information. And there's a proliferation of personal care products containing PFAS, and our understanding of this sort of relative importance for the general human population is limited. And so when I've been part of a number of review panels now where we're asked to say, what do we understand about the predominance of any given PFAS exposure source for different human populations? And the one thing we can say that we do know is that drinking water is the primary pathway of PFAS exposure next to contaminated sites. And what I'm showing you is a visualization of contaminated communities from the Northeastern Initiative, so Phil Brown's work. And it shows, our understanding of these contaminated sites is really growing as we detect more and more. There are different, the diversity of types of contaminated sites is expanding from not just manufacturing locations and industry into these Air Force bases and fire training areas where they use the aqueous film forming foams containing PFAS, and now even some landfills and other sources. So next to these sites, then when we think about exposure, we really think about drinking water. And I believe that's actually biased our research a little bit. Of course, this is a hugely important topic, but what it's meant is that we don't necessarily understand, this looks very similar to Jackie's source. I'm not sure why the animation there isn't working. So good that Jackie showed us that. This was meant to show, if you can re-envision her picture of all those different products containing PFAS. That's what this slide was meant to show. So it's not just these single source, it's all these diverse pathways that I showed you a moment ago. And so for contaminated communities, we have a good handle on the predominant exposure pathway. For the general population, we really have to think about the diversity of those sources. This also becomes important when an individual in a contaminated community or with a contaminated drinking water supply, and that drinking water supply starts to be remediated or cleaned up, then often individuals want to know, well, what are the other PFAS sources that I'm receiving that I should be thinking about? And that's where we have to start thinking about the other dietary sources, the consumer products, and the seafood sources as well. So we've thought a little bit about these drinking water pathways for the general population. This is a paper published several years ago now. And this is thinking about it on a federal scale. I'm sure many of you have seen this diagram over and over again. And the main point of this is really, that even for the general population, the likelihood of detecting PFAS in your drinking water supply can be predicted by abundance of different known PFAS sources. And so in this paper, we looked at industrial sites, military fire training areas, AFFF certified airports, and wastewater treatment plants. So the number of those sources within a hydrologic unit is directly associated with the probability of detecting PFAS in your water supply. And we've since, through a variety of other work that I won't show you today, refined that to include many other sources. So we can kind of have a good sense of where drinking water supplies are likely to be vulnerable, even outside of what we would define as one of those major contaminated communities. The next question we ask then is, for the general population, so this is outside of those contaminated communities, how important is drinking water as a vector? And this is a study we did in a longitudinal cohort of US women that's very similar to the general US female population. So it's comparable to the NHANES survey, the National Health and Nutrition Exam Survey data for women of this time period. And we were looking in this case at paired water and plasma samples to understand how important is drinking water for that general population of US women. The collection, so this is sort of a unique cohort because they collected tap water from the individuals at the same time as they collected the blood. I'm just trying to give you the highest level message from this study, but it provided a unique opportunity to look at the contribution of drinking water in the general population. And that's what I'm showing you here is the association between serum concentrations and water measured in the tap water. And I'm showing you a couple examples. You can look at the paper for all the different ones. I've now corrected for that one high leverage individual for PFOA, the first compound you were looking at. But you do see statistically significant associations, even at much lower levels than you would find in a contaminated community, between tap water PFAS and serum concentrations of PFAS, and it's stratified by the amount of tap water an individual reports consuming. And overall, we find this is the range across the compounds we were able to look at, accounts for between 2 and 34% of overall exposure determined by measuring these compounds in the serum of these individuals. The regulatory guidance for drinking water uses something called a relative source contribution of drinking water of around 20%. And we find that our estimates from this cohort agree fairly well with that. So that gives us a first indication of the relative importance of drinking water relative to all those other diverse sources in the general population. Now, the collection that took place in that cohort, the paired serum and tap water, was actually quite a while ago. So it was 1989, 1990. So those were the data you were looking at. And of course, we know that PFAS production has changed quite dramatically since that time. So we conducted a pilot study just going back to some of those public water supplies for those individuals and looking at how the composition of PFAS in those water supplies has changed over time. And this touches again on a message that Jackie provided nicely in the introduction, which is we have many, many PFAS now that we know exist. A number of them, or we know these PFAS are registered with the OECD. Hundreds of them are in commerce, as EPA said. And so this, you can think our methods for analyzing them target just a few of those compounds. And I don't want to bore everyone with an analytical chemistry lecture, but this actually becomes really important to how we think about this field and how to move forward, in that the health studies and the exposure studies are typically studying this what we call targeted PFAS. So just a few of the PFAS compounds. Right now we have commercially available analytical standards for maybe 40 of these compounds. And we know that there's this much larger universe of compounds being produced and used, and we're sort of missing that. So what we found in this study, using a proxy for total organofluorine that I'm going to call EOF for the rest of this talk, so it's extractable organofluorine. If we go back to looking at what's happening in the drinking water, we see the change in those targeted PFAS, which typically are the old ones that we've produced for a long time. So we have a commercially available analytical standard. There's no large change apparent between 1989, 1990, and 2016 in these water supplies when we collected this sample. But the fraction of unidentified PFAS or new PFAS increase quite dramatically. So this is something that we really need to think about and study moving forward. The next thing I wanted to just briefly touch on is the thinking about, well, how do we start to understand these major sources when we don't have, good dietary survey information, and we don't have, all the data we might need in every population to think about dominant human exposure sources. So one of the things that my group has been playing around with is looking at the composition of these compounds in human blood. So if you do targeted analysis, you can actually detect, as I mentioned, up to 30 compounds. And those compounds in themselves provide some clues as to major exposure sources. One of the ones that we find human blood provides a particularly good tracer for is for seafood, because the most bioaccumulative PFAS are the longest chained legacy compounds, so the PFSAs. And so and the carboxylates, the long chain carboxylates as well. So we find the C11 compound, so this is a aliphatic chain with 11 perfluorinated carbons, is a particularly good tracer for seafood consumption. And we've looked at this both in the NHANES survey and in the Faroe Islands children. So this is the serum level of this compound. And we've correlated in this case with hair mercury, because the predominant exposure pathway for mercury is seafood consumption. And you can see this very nice association. You can also see this emerge in NHANES. So the high level message is that I think we can use the human serum data more than we have to date to actually provide clues as to major exposure pathways, and particularly for things like seafood. Now, the Faroe Islands are a traditional whaling population, so we expect them to be on the high end of seafood exposures. And I want to transition now into thinking about consumer products as I say this, because part of our analyses in this population included a characterization of the contribution of seafood to overall exposure of these children in the Faroe Islands over time. And one of the surprising findings for us was that that contribution from seafood has remained relatively steady, maybe increased a little bit in recent years, but the serum levels in the children actually declined quite dramatically post 2000 when there was a voluntary phase out in the production of the parent chemical to PFAS and its precursors in North America. So this is very interesting. We see this rapid decline in serum that can't be explained by seafood in this high seafood consuming population. We know that drinking water in this population is below detection for most PFAS. And so this is very suggestive of, even in this remote island population, that consumer products play a large role in overall exposure. And so we've done a little bit of work on this, and many other groups have done work on this, showing that in consumer products, there are really, really large fractions of unidentified precursors. So it goes back to that analytical chemistry story, which is where we're missing through standard techniques for measuring PFAS, the majority of substances to which we're exposed in consumer products. So this is definitely an area of future work, trying to understand what that means for our exposures. I think that's generally true for all these different exposure vectors. For consumer products, I'd like us to follow the European model a little bit more, saying there are substitutes that work well and perhaps we don't need them in our products after all. I want to switch gears a little bit in the few minutes I have left to talk about the global scale then use of PFAS and what this means for individual exposures and our understanding. So we're trying to link this idea of chemical production and use on these different spatial scales to individual exposures. And when you start to think beyond the individual source, you can start to think about the diversity of these different sources. And one thing that we've really missed in the conversation and only just started to skim the surface on is this atmospheric transport pathway that you can see. And this is why we detect PFAS in these remote regions, in addition to long-range oceanic transport. But we're really just starting to work on this. And I really think that atmospheric transport has been overlooked as a potentially significant source next to many of our communities where we have a variety of PFAS sources as well. So this is something that represents a major gap. In my research group, we've started to try to develop these source receptor relationships to better characterize how significant atmospheric inputs are to different regions and how important they are. We're using a global model that we use for many different chemicals at Harvard. This is called GeosChem. And one of the challenges right now is really data on releases, on deposition, in ambient air. There are a few papers starting to come out, and I know the regulatory agencies are just starting to think about that. So this is, in my opinion, very important work to keep moving. As we have these measurements, we'll be able to better develop these tools to characterize the importance of this atmospheric transport pathway. We've done a little bit more on rivers, and this is predominantly for PFAS or PFAS. And sorry, I didn't mean to skip there. And for PFAS, it's an interesting one, because when we think about it, I put this in because the second part of my title here was global sources, but there's been a shift in these sources for PFAS from predominantly North America, which is why it's colored the dark green here. So this is historic discharge and the intensity of historic discharge. And this is work by my doctoral student, Charlotte Wagner, where she's looked cumulatively at PFAS releases from global rivers. And these circles are showing you the magnitudes of current production. So you see that North America is the predominant legacy source, but now those sources have shifted to South America and Asia. And we can look at what that means for the global ocean. So I want to end on what this means, and also, if I can, which is difficult with these compounds, but end on a slightly more upbeat note, which is for PFAS and PFOA, we've actually taken quite stringent global action. And if we look at the impacts of those global actions, we can first think about, and so this example is for PFAS, we can first think about how that impacted seawater, which is the terminal sink for these compounds in North America. This is a modeling study that we published a few years ago. But the peak production occurred around the year 2000. And so you see the ramp up to peak production, and then those compounds were voluntarily phased out in North America. And you see a relatively rapid decline in concentrations following the phase out of that production. You can see that same decline if you use marine organisms like the pilot whale as a species, sentinel species for that change. So we looked at pilot whales over different time periods. And again, this builds my atmospheric story. So you don't necessarily see it in the PFAS compound itself, but the precursor to PFAS shows this large decline and rapid decline. And we also see that in the children. So the whales don't metabolize that precursor and turn it into PFAS. But the children do, so that foci I just showed you turns into PFAS, and we expect that drives, and you can see that in the individual compound. These are children in the Faroe Islands, this rapid decline. And that rapid decline in serum for PFAS has been seen in many different populations. So I would say that we have lots of challenges ahead of us in terms of mitigating risks associated with these compounds. And we also have evidence that when we take, united global action to address this problem, then we can have success on very rapid timescales at reducing these exposures in some of these vulnerable groups like children. So if I could summarize my various points today, I hope you come away with this idea that human exposure sources for PFAS are diverse. We have the most data on drinking water. There are important gaps still remaining in drinking water, but we have more data on drinking water than other sources, such as dietary sources, consumer products, and seafood. And so these are all things that need to be addressed. The importance of PFAS precursors that degrade into the terminal PFAS that, like PFAS and PFOA, that have been associated with some of these concerning health impacts, are things that we need to really better understand. And this is very much an analytical chemistry issue right now. So we have these standard methods that are limited by the availability of commercial standards to run our instruments. And so we really need to think in an innovative way about other ways to measure these newer PFAS that are being produced, because we're not able presently to keep pace with the production of new PFAS using those standard measurement methods. And then on a global scale, I think, and again, this is my opinion, but I think that this atmospheric transport pathway is really, really important, and we have only preliminary data on it. We do have data on remote environments like the Arctic and Antarctic, where we see high levels and suggestive data next to large point sources that this is a really important pathway. And we're at the point where we really need more measurements of this source. So I'll stop there and turn it back to Jackie, and look forward to questions at the end. Thank you.
Yes, thank you very much, Dr. Sunderland, on that great presentation, and especially on ending with a hopeful note too about the state of PFAS. I have a quick housekeeping item that's been brought to my attention. If you're on the phone and not, on a computer or something like that, you might not be able to use the Q&A. So if you have a question and you use the raise hand function, I think I can allow you to speak and ask that question later on during the Q&A session. Okay, so our next speaker is Dr. Jamie DeWitt. Dr. DeWitt is an associate professor in pharmacology and toxicology at East Carolina University. She's also a diplomat in the American Board of Toxicology. She completed a PhD in environmental science and neuroscience at Indiana University in Bloomington, and then completed some postdoctoral training in immunotoxicology, after which she joined the faculty at East Carolina. Her research program explores how early life exposure to a variety of toxic agents, including various PFAS, may impact the immune, nervous, and endocrine systems, and how this can result in altered development and effects on children's health. Today, Dr. DeWitt will, of course, be presenting on the immune effects from PFAS exposures.
Well, thank you so much. It's really my pleasure to be here, and I appreciate being part of this mini symposium. For those of you who attended the Michigan Society of Toxicology meetings, you might see some slides that are a bit familiar because I may have recycled some of those slides, but I have some new information to present. And for those of you who weren't able to attend this meeting, thank you for being here. It'll be all new for you. I just want to share real quickly that I may have some bias. I do some expert consulting work for legal firms. I speak publicly about PFAS, and I advocate for communities who have been exposed and are being exposed to PFAS. So that may introduce some bias into how I perceive the toxicity of these compounds. But first, let's talk a little bit about why the toxicity of PFAS are a concern. We've heard about the exposure, and we know about human and wildlife exposure and concentrations of PFAS in environmental media, but why are we concerned about these chemicals in the environment and in our bodies? Well, first, we have many, many different subtypes of PFAS. We've heard the number 2,000, 800, 4,000. I have 8,000 on my page. This is a recent estimate from the United States Environmental Protection Agency based on the chemical structure. Most of what we know about PFAS, however, as Dr. Sunderland indicated, comes from a relatively small number of this very large class of substances. And most of the toxicological information comes from the perfluoroalkyl acids, the compounds like PFOA and PFOS. But as Dr. Sunderland indicated, we are likely exposed to many different combinations of different PFAS, and where you live may influence your exposure profile and make you more exposed to PFAS from AFFF versus those from industrial facilities. And one of the toxicological concerns that we have about PFAS is associated with their chemical structure, that persistence. The persistence of PFAS really drives a lot of our concerns about these compounds. And you've heard the term forever chemical. That term is really associated with the strength of that carbon-fluorine bond. So all PFAS, except for those that maybe transform, but remember they transform to persistent end products, are persistent. We understand that many PFAS can bioaccumulate from the environment into living organisms. We know that many PFAS also are mobile so that they can move from sites distant from their points of release. And we also understand that those PFAS studied have exhibited some form of toxicity. We've seen some information already on some of the concentrations in exposed wildlife. What I present here are some maximum PFAS, or perfluorooctane sulfonate, concentrations in various organisms. And this is an illustration of how PFAS and other PFAS can accumulate into living organisms from environmental media. Some of these organisms are important to us because of the ecological services they provide. Some of these organisms are important as food sources. So not only do we lose perhaps diversity when organisms suffer toxicity from their exposure, but we may lose sources of food and increase our exposure to those sources of food that have PFAS contamination within them. We also know that PFAS can accumulate into our bodies because of efforts on the global scale to phase out the production of PFOA and PFOS in many types of processes. We have seen declines in concentrations of PFOA and PFOS in human blood across time, but people who live in contaminated communities with point sources of exposure and people who work with PFAS can still have very high concentrations of PFAS in their bodies compared to other people people who are exposed through different sources. We also don't measure for the vast majority of PFAS. So again, as Dr. Sunderland highlighted, many PFAS that are in the environment are unknown quantities inside of our bodies because we haven't measured for them inside of our bodies yet. And the issue with persistence then is related to the very long half-lives of many of these compounds. If you look along the bottom of your screen, you can see the estimates or known human half-lives for some of the different subtypes of the perfluoroalkyl acids. They're arranged from shorter chain to longer chain, and for the most part, PFAS with a higher number of carbons take a longer amount of time to get excreted from our bodies. And that's one of the reasons that we focus so much of our efforts on the compounds that we call long chain PFAS. But if you look at some of the shorter chains, those with 4 carbons or 5 carbons, you can see that the half-life from the perspective of a pharmacological agent is still fairly long. Imagine if you took an aspirin and you couldn't take another aspirin for 3 or 4 more days because of its concentrations inside of your body. So 3 days from a relative perspective is still fairly long. Now, certainly it's short when you compare it to 8 years, but from the perspective of an agent that we really don't want in our bodies, 3 days is a fairly long half-life for some. I wanted to highlight some of the general toxicological findings that have been found for PFAS. A lot of the findings that we understand come from the C8 Science Panel study that was already alluded to. This was a study of a large number of people living in the mid-Ohio Valley area. Approximately 70,000 people participated in this study, and not all were studied for all endpoints. But the 3 epidemiologists who navigated this study determined probable links for PFOA in this particular community. And you can see them listed here. 2 types of cancer, elevated cholesterol, pregnancy-induced complications, thyroid disease, and ulcerative colitis. And if any of you have been following some of the popular press about PFAS, you know that the class action lawsuit that resulted in this epidemiological study was featured in the movie Dark Waters, and it has been aired fairly recently. But if we look at a greater number of PFAS beyond just PFOA, we can see that additional types of health outcomes have been associated with people who have been studied and whose blood has been measured for concentrations of various PFAS. The Agency for Toxic Substances and Disease Registry evaluated 14 different PFAS, and in 2018 published their toxicological profile. And you can see that there are many different types of health effects, which tells us that PFAS have the potential to perturb many different organ systems in our bodies. For example, some of these are endpoints that can affect developing organisms. So we see changes to the pregnant mother in terms of hypertension and preeclampsia. We've seen decreases in birth weight. And then we've also seen changes to immune responses. And these changes in immune responses have been observed both in children and in adults. And I'm going to focus the rest of my talk on these immunotoxicological responses. A recent paper came out from the epidemiologists and some of their colleagues who were part of the original C8 Science Panel, and they evaluated some of the additional studies that have been published regarding the associations of PFAS and some of the health outcomes that they identified as probable links. And they noted that many of these different types of health effects have not really grown in terms of the strength of their associations, but it's not because we have negative data indicating that these associations are really weaker than we originally thought. It's just that we don't have the additional data to give strength to those associations. So it's absence of evidence rather than evidence of absence. But I really want to highlight these alterations in immune responses. We have very good evidence that exposure to PFAS is a concern for both the adult and the developing immune system. And I really want to then take us into talking about how we can work with what we understand about PFAS toxicity to start to protect public health. And one of the challenges from a toxicological perspective is that PFAS do affect many different systems in our bodies. So how can we best protect public health? Protecting developing organisms has been recognized as a priority concern. And if you look at some of the health protective measures that various states have taken and that the US EPA has taken, the emphasis is on protecting developing organisms. I would like to argue that the immune system can be just as protective as some of the health effects that have been based on developmental toxicity. So as I mentioned, alterations of immune responses is a piece of epidemiological evidence that has been strengthened by additional epidemiological studies. So I'm going to talk specifically now about the immunotoxicity of PFAS. And let me define for you how I view the world of immunotoxicity. And I view the world of immunotoxicity from the endpoint of immune function, or from the aspect of immune function. In order to understand whether or not the immune system is doing what it's supposed to be doing, you have to ask it to do something. So you have to challenge it. And one of the ways that we challenge it here in the laboratory is by inducing a vaccination. So if you give the immune system a vaccination, it should develop protective antibodies against that vaccination. If you see failure to develop protective antibodies, then you know that the immune system is suppressed in some way. Suppression of the immune system can increase the risk of infectious diseases. It also can increase the risk of certain types of cancers because our immune system plays a very important role in cancer cell surveillance. On the other hand, if you ask the immune system to do something and it goes way over and above, that's hyperstimulation. And one of the endpoints that we can use to understand hyperstimulation is the presence of allergy and asthma, or perhaps autoimmune disease. Somewhere in the middle is inflammation, and inflammation is a necessary part of the healing process, but if it goes on for too long or is too robust, it can lead to disease pathologies. So I'm going to focus specifically on some of the endpoints that have been associated with responses or aberrant responses to vaccines. And I'm going to present to you a little bit of information from a report that came out in 2016, and they used a weight of evidence approach. In other words, the report that I'm going to share with you looked at all of the evidence available for understanding effects of PFAS on the immune system and made a presumption about whether or not PFOA and PFOS were hazardous to the immune systems of humans. This agency was the National Toxicology Program, and in 2016, they published a systematic review of the literature associated with PFOA and PFOS and their potential to affect the immune system. The National Toxicology Program evaluated this weight of evidence and determined that because both PFOA and PFOS are able to suppress this vaccine response, this antigen-specific antibody response, that they were presumed to be immune hazards to humans. So there's evidence from experimental animal models, and there's evidence from epidemiological studies of humans. So we can accept then, based on this weight of evidence, that both PFOA and PFOS are hazardous to the human immune system. And what's really important about this particular systematic review is that the National Toxicology Program prioritized immune outcomes based on those functional outcomes. So they looked specifically at studies that evaluated whether or not the immune system was rising to a challenge or rising too much to the challenge. So they measured these functional immune or primary outcomes. Secondary immune outcomes, such as measurement of cytokines or looking at weights of different lymphoid organs, were important in supporting their primary determination. But those functional outcomes really drove the decision. And that's important because these functional outcomes tell us whether or not the immune system is really doing what it's supposed to be doing. So let me tell you a little bit about suppression of antibody production. It's a fairly complex process. It involves multiple different cells, but some of the work in my laboratory has highlighted or uncovered that B cells, the cells that eventually transform to make antibodies, are the cells that are impacted by PFAS. So on the left, I have some graphs of some of our studies with PFOA. The top graph illustrates something known as the T cell dependent antibody response, and the graph on the bottom is the T cell independent antibody response. The difference between the two graphs is that the graph on the top requires that T cells go to B cells and say, hey, B cell, make this antibody. Whereas the graph on the bottom highlights that B cells recognize the antigen and could make antibodies on their own. So this, because we saw suppression in both of these endpoints, we knew that B cells are targeted. And when you see suppression of B cells, you can see reductions in the vaccine response, and you also can see an increased risk of infectious diseases. In terms of protection of public health, immunotoxicity has been incorporated in some public health protections across the United States, as well as in European Union countries. The immune system is a sensitive health endpoint, and it can be health protective of individuals exposed to PFAS, especially through their drinking water. But with the last few minutes that I have left, I'd like to talk a little bit about some of the emerging PFAS or understudied PFAS that we see here in North Carolina. In North Carolina, one of our watersheds, known as the Cape Fear River watershed, has been contaminated with PFAS from a fluoro production facility, and it's depicted here on the map. And in 2016, a group of researchers from the EPA and NC State and some of our UNC system schools looked at the water in the Cape Fear River and measured different concentrations of PFAS. And they used a non-targeted approach. So they deduced structure from the signals that they were measuring in their instruments. And in this particular figure, you can see that there were many different types of PFAS in the water that were previously unidentified, so understudied. And community C, which was the community downstream from the fluoro production facility, had very high concentrations of a compound that we found out later was known by the trade name of Gen X. In 2017, a local reporter published an article in the Wilmington Star News, and thus began North Carolina's journey into uncovering the different types of PFAS in our environment and what they do to our health. So I'd like to share with you a little bit about the approach that we're taking in my laboratory with funds from our North Carolina General Assembly through the North Carolina Policy Collaboratory to study these understudied PFAS. So we've divided our research into two phases. And in phase one, we studied some of the compounds that were listed in a consent order between the state and the facility that manufactured these compounds. And in phase two, we're going to start evaluating some of the compounds that have been uncovered through some additional teams in our PFAS testing network. We should have publications for some of the chemicals in the first phase coming out fairly soon as my student and postdoc give me their next round of edits. So this is our general toxicological approach. This is a descriptive toxicological study. We're evaluating the effects of a 30-day exposure period in mice who are given these compounds in their drinking water. We collect urine and feces to understand not only concentrations of PFAS in urine, but also the effects on the microbiome, and those effects are being evaluated by a collaborator. We give vaccinations during the exposure period. And then at the end of the study, we ask questions about the immune system as well as some other general toxicological endpoints. These studies are being driven by my postdoc, Dr. Tracy Woodliff, and then by Emma Tobin, who's a master's student, and Sam Vance, who is now an ORISE fellow at the US EPA. And in terms of the compounds that I'll highlight here, I wanted to focus specifically on PFMOAA. This is a 3-carbon compound that was detected in high or relatively high concentrations in the Cape Fear River. And I'm going to contrast that with Naphion byproduct 2, which is a 7-carbon compound that was detected in fairly low concentrations. So we have differences between these compounds in terms of their number of carbons and their number of oxygens. So these are perfluoroether carboxylic acid or sulfonic acid compounds. So the PFMOAA is short and has 1 oxygen. The Naphion byproduct 2 is long and has 2 oxygens. What I have here are weights of livers. We often look at liver weight as an important endpoint of PFAS toxicity. Many PFAS are known to increase liver weight in rodent models. And if you look at PFMOAA across the top, we see that there was not an effective PFMOAA exposure on liver weight, but our PFOA positive control still increased that liver weight. If we look at Naphion byproduct 2 in the bottom graphs, we see that there is a dramatic increase in liver weight in our highest dose group, which was 5 milligrams per kilogram. weights of the livers in these animals were increased by about 300%, whereas PFOA only increased the weight by about 200%. I'd also like to point out that Nafion byproduct 2 had impacts on the weights of the spleen and the thymus. PFOA did not, and we didn't see any changes in body weight, so there are no signs of overt or systemic toxicity in this 30-day exposure period. If we look at the TDAR, or that T-cell dependent antibody response, or the response of these animals to their vaccination, we can see that with PFMOAA, we didn't see a statistical decrease at any of our administered doses. We did see a decrease from the concentration of PFOA that we gave as our positive control. If we look at Nafion byproduct 2, we see a profound reduction in antibody responses in that 5 milligram per kilogram dose group. So what this suggests to us is that PFMOAA is less potent, possibly due to its chain length, compared to Nafion byproduct 2, which is a longer compound and has 2 oxygens. I'd also like to point out that when we evaluated serum concentrations, we couldn't detect PFMOAA in the mice, but the concentrations of Nafion byproduct 2 were quite high, in the hundreds and thousands of nanograms per mil or parts per trillion. So a few final thoughts before my time is up. Where do we go from here? I know that Dr. Sliver is going to be talking about the response to PFAS in Michigan from a policy perspective, but I wanted to just bring in a touch of policy to help to transition. We have many different types of PFAS in the environment. We're still trying to figure out where they are. We're still trying to figure out what they do to living organisms. What do we do? Should we use my approach and address PFAS one by one? Well, if we have 8,000 different substances to address, it's going to take us many, many lifetimes to understand everything that we need to understand about these compounds if we want to regulate or manage them based on a one-by-one approach. But a couple of papers have come out this year suggesting that some different approaches might be warranted for protecting public health from PFAS. One paper by Kwiatkowski et al. Suggested that we need to manage PFAS as an entire class, and another paper by Cousins et al. Evaluated different types of grouping approaches that have been used across different European member states and across different states here in the United States. The PFAS as a class approach is based on the concept that all PFAS are persistent, many bioaccumulate, many are mobile, and many are toxic. And so we need to apply these principles to all of them to effectively protect public health. In terms of different approaches, most states and countries have used a least precautionary approach. So they've grouped compounds based on similar toxicity. A more precautionary approach would be grouping compounds based on persistence. With that, I'd like to acknowledge my PFAS funding and all of the hard work of the people in my laboratory and say thank you, and I look forward to the Q&A session later. Thank you for listening.
Thank you very much, Dr. DeWitt. That was a great presentation on some of what's known so far about immunotoxicity and PFAS. And I appreciate the notes about how to regulate groups of them as well. And that's probably something we can talk about during our Q&A. So for the final speaker of today, we're going to switch gears and talk about how science is used in shaping policy and programs with, the state of Michigan as an example. So I'm pleased to introduce Steve Sliver, the Executive Director of the Michigan PFAS Action Response Team. This is Michigan's unique multi-agency approach to address PFAS contamination across the state. Steve was trained as an environmental engineer at Michigan Tech University, and since then, he served the state of Michigan for over 30 years in various capacities, formerly the Assistant Director of the Material Management Division for EGLE, which is the state's Department of Environment, Great Lakes, and Energy. He's now the director of this really important MPART group. Under his guidance, Michigan is becoming a leader in how PFAS policy and management are done, and we have the pleasure of hearing about that today.
Hey, thank you very much for that fine introduction. I really appreciate the opportunity to be here today and share, how Michigan is moving ahead and with our data-driven, science-based approach to protecting the public from PFAS contamination. And I really appreciated the previous presentations that really kind of set the stage for why we're doing what we're doing here in Michigan. And from those previous presentations, we know how pervasive and how persistent PFAS are. And this conceptual site diagram really illustrates how we are cycling PFAS throughout the ecosystem because we're not destroying or sequestering them. And I always like to use this slide to show, like, for example, the landfill as an example, where all of those consumer products and industrial waste end up there and release PFAS into the leachate, which typically is not treated for PFAS. And so when it goes on to a wastewater treatment plant that doesn't treat for it either, much of it passes through into our surface waters, impacting potential drinking water sources and fish. And some of those sludges generated by the wastewater treatment plant actually go back to the landfill. Perfect cycle, right? And then some of them are land applied as biosolids, where they could be impacting, agricultural fields, soils, groundwater, surface water runoff, and maybe even crops with some of the PFAS that are accumulated in those biosolids. So really, our challenge is to understand the occurrence of PFAS throughout the cycle so that we can break it and protect public health. And, Michigan's confronted this challenge by creating the Michigan PFAS Action Response Team, or MPART, and charging it with planning and implementing all state actions relative to PFAS. It's an advisory body comprised of agencies responsible for environmental and natural resources protection, public health, agriculture, military installations, commercial airports, and fire departments. It really breaks down those silos of bureaucracy to help ensure that we've got an effective and efficient response. And it's also responsible to coordinate with the federal and local government, as well as other stakeholders, to leverage all of our resources for common goals of protecting public health and the environment. I think, MPART really is the reason Michigan has been so successful at rapidly identifying and reducing exposures to PFAS contamination with our data-driven, science-based approach. And policy, is derived from the legislature too, not just the executive. And we've enjoyed great legislative support over the past couple of years. PFAS contamination does not respect political boundaries and parties. And we've been very fortunate to have that bipartisan, bicameral support. We've had significant appropriations over the past few years, over $70 million since 2018 to do the kind of response that the governor has asked us to do. And they passed legislation which limits the use of the, Class B AFFF that contains PFAS and requires, fire departments that use it to report it to us so we can make sure that environmental impacts are minimized. And we just concluded a AFFF collection and disposal program, probably the largest in the country, where we collected over 51,000 gallons of this material from fire departments across the state that didn't want it or need it, and basically getting that out of the system as a pollution prevention effort going forward. And many of you may have seen the governor just announced $25 million in grants to aid municipal water systems that are impacted by PFAS contamination. So MPART's response has been very comprehensive, and priority is protecting public health. And we've had to do everything from work on developing sampling and analytical methods, studying where this stuff is, what the sources are, what the environmental fate and transport is, health effects, all the way to better understanding, how best to regulate this material going forward. The primary pathway of concern, as you've heard, for exposure is drinking water. And so Michigan is really taking action in a very deliberate way to protect the public's water, drinking water and its sources. And we started with public water supplies. EPA came out with their lifetime health advisory of 70 parts per trillion of PFOA and PFOS. I think it was back in 2016, but no one's really required to monitor or to address any results that they might get if they do monitor. We wanted to understand what's in our drinking water. So we tested all community water supplies, and the systems for schools and larger daycares on their own wells. This really covered 75% of Michigan's population. And since we began that back in 2018, we've added additional supplies, smaller supplies of the more vulnerable populations, and gone back to better understand some of the concentrations that we found in those systems from the earlier testing, and whether or not, for example, surface water intakes would see fluctuations in any levels that we had found. This is really just an oversimplification of the results that we received over the past couple of years. And again, this represents water for 75% of Michigan's population. It doesn't address the 1 million private residential wells that serve a quarter of Michiganders. But you can see here that about 90% of those systems didn't have reportable concentrations of those 14 PFAS analyzed under EPA's method, that nationally validated method. Another 7% had something less than a total of 10 parts per trillion for all 14 of those compounds if you added them all up. And that's something that MPART determined early on, back when these surveys began, that we probably didn't need to do much with those results other than continue to monitor to make sure they don't go up over time. 3% had something more than that 10 parts per trillion total, but less than 70 parts per trillion PFOA and PFOS. Those are those supplies that we've gone back and been doing quarterly monitoring, working with them to help determine if there's sources in the area and what options they might have if, in fact, standards were imposed, enforceable standards, and or if the levels went up. And unfortunately, or fortunately, we found 3 systems that exceeded that 70 parts per trillion, including the city of Parchment and Cooper Township, serving over 3,100 people that had concentrations in the 1,600 parts per trillion range, and as you can imagine, required an immediate response. And you know, all of the results that were gathered from this survey have been transmitted back to the supplies, and with recommendations based upon that 70 parts per trillion, and additionally notifying their customers and monitoring long-term, looking for alternate sources if necessary and what their options might be. But you know, we try to be data-driven here in Michigan, and you know, I think finding the concentrations of PFAS in the water supplies, really covering over 500,000 Michiganders, it was clear we needed to operationalize the ongoing monitoring and addressing PFAS contamination in our drinking water. And typically we do that through standards that are developed by EPA at the national level that we then integrate into our program. But unfortunately, EPA, while they have a plan to develop standards, they're likely only going to get a couple in place over the next several years. And we had a report from a science advisory panel back in 2018 that says, you know, that 70 parts per trillion is probably too high, and you should be looking for more than just PFOA and PFOS. And that's why Governor Whitmer back in March of last year directed MPART to move forward with facilitating the development of our own state-specific MCLs, or drinking water standards for PFAS. And it began with a science advisory work group to give us some health-based values as a starting point, and then EGLE moving forward with developing the actual rules. And so we charged the science advisory work group with looking at all of these proposed and existing standards and the basis for them across the country, you know, what they were based upon, you know, all of the different factors that go into these risk assessments, risk evaluations, and make recommendations back to MPART on health-based values for any of those PFAS compounds that were evaluated under the newer EPA method, 18 compounds, where there was enough peer-reviewed, defensible studies out there in the literature that they could support moving forward with drinking water standards for PFAS. Now, they gave MPART that report. It was a very comprehensive, well-done report that has been so critical for us moving forward here in Michigan with our drinking water standards. They came back with 7 recommended PFAS levels or recommendations for 7 compounds. And they also recommended at the time that based upon the science that was available, that we ought to be looking at regulating them individually in drinking water systems, and also recommended that we use as a screening value the 6 parts per trillion for any of the other compounds that we didn't have a specific value coming out of this report. And we're very fortunate to have Dr. DeWitt on that panel. So thank you again, Jamie. And so we adopted the report, recommended that EGLE move forward with developing rules. And this is really where kind of science meets some of the regulatory policy development. And they started with the health-based values, and then had to consider a number of other issues, get stakeholder input, actually go before a kind of what's called the Environmental Rules Review Committee, an oversight committee for development of environmental rules, and then ultimately the legislature. And just give you an idea what some of those other considerations for rules could be. Basically, it's how feasible is it to move forward with those health-based values? Can you analyze for them? How effectively can you treat it? And what's it going to cost? What is the implementation time frame that you're going to impose on the supplies that will have to meet these standards? Well, ultimately, we went through with the rulemaking, and on August 3rd, the 7 standards became effective in Michigan. We didn't adjust any of those concentrations that came out of the original recommendations from our Science Advisory Work Group. Just a couple of things to highlight here. As you can see, number one, PFOA and PFOS significantly different than the 70 parts per trillion recommendation from EPA, as well as the compounds themselves, and the 6 parts per trillion versus 420 or 400,000 parts per trillion, based upon where the science led the development of these standards. And now we have 2,700 water systems who are going to be moving forward with implementing these requirements. I should note that those MCLs did not translate directly into cleanup criteria for groundwater. We had criteria for PFOA and PFOS based upon that EPA lifetime health advisory. By rule, we now have those 2 criteria from the MCLs automatically. Those now become our new criteria for groundwater. But the other 5 PFAS compounds, before they become effective as groundwater cleanup criteria, will also have to go through a rulemaking process. But it's also very important for how we define sites, because many of you have heard how many sites Michigan has and how contaminated Michigan must be. Well, number one, you've heard me say this before, it's not because we're more contaminated, it's because we're looking more rapidly and comprehensively than anyone else. And we define a PFAS site as one that exceeds our criteria. So as you can see, on August 3rd, when those MCLs became effective, the number of sites that we'd identified with lower concentrations bumped up our site numbers from 99 to 138. But these are landfills, military installations, old industrial sites, as well as commercial airports, places where that AFFF was used. And we prioritized every one of those investigations based upon whether or not we had any concerns about contamination migrating off-site and impacting private residential wells or other drinking water uses of those aquifer. And when we do identify a site, this is when all of MPART comes into a community and trying to be as transparent as we can, identifying what we have uncovered, what we're going to do about it, and what the results that we get along the way might mean. And at a minimum, anytime that we find a significant exposure or threat of drinking water, we have alternate water provided in these communities, and an ongoing transparent approach to keep the residents informed while we're doing the investigation work. Again, drinking water pathway is the primary pathway of concern here. But we've also integrated PFAS testing into our surveys of surface water and fish. That enables us to track down sources of contamination into our waterways, and also identify fish where we might need to issue a fish consumption advisory based upon accumulations in them. And surface water foams, we found really high concentrations of PFAS in surface water foams on impacted water bodies. And now going forward, when we cite this foam that, it looks pretty unique, that will help inform where we move our surveys of these waterways going forward. And we've asked our wastewater treatment plants to work with their users to reduce pollutant loads coming into the wastewater treatment plant so that we don't have to worry about something passing through into our waterways or impacting biosolids. And that's the Industrial Pretreatment Program initiative that began back in February of 2018. And it's a great example of success where you've got state agencies, local agencies, and the industrial users working together to reduce pollutant loads so that they don't get through into our waterways and biosolids. And it's also produced some studies which are very helpful going forward from a pollution prevention side and helping us understand where we should be targeting our resources. Fume suppressant study was done with EPA's Office of Research and Development, looking at what fume suppressants contain. Those are used by metal platers, metal finishers, to protect the workers, and historically have contained PFAS. And so a study was done to see if there's PFAS in any of these ones that are currently being used, as well as we have the studies of what are the major sources of PFAS in our wastewater treatment plant systems. And, whether it's landfills, central waste treaters, metal platers, et cetera. And also those studies have shown us which biosolids seem to be more impacted than others, and giving us some screening criteria on what's industrial impacted, where we might not want to land apply it until we better understand, crop uptake and other impacts on the environment. And I will say Michigan is fortunate from a policy perspective that we have criteria for surface water. And we have 12 parts per trillion, 11 if it is a source of drinking water for PFOS, and then 12,000 parts per trillion, 420 if it is a drinking water source for PFOA. And those values have enabled us to drive forward on our investigations and reducing pollutant loads into our systems. And trying to address PFAS, whether it's in drinking water, wastewater, or groundwater contamination, has really revealed to us that we need more options to address the contamination. And so that prompted the development of our treatment technologies roundtable last May. And first and foremost, just to create a network of all of the people who have information to share about what technologies are available and what are the best applications of them, and what are our research needs going forward, and then to coordinate on those where we can so that we move the ball further down the field than we would individually. And they identified some research gaps, whether it's in rapid detection technology, destruction technology, and the ability to better treat contaminated drinking water, that in those technologies that might also translate into other wastewater streams. Also, this group wanted to make sure that we put on a training session, and there will be one now at the end of October. It's the Great Lakes PFAS Summit, the whole last week of October of this year, this month, right? And where we're going to be bringing together experts, everything from the health effects all the way up to treatment technologies on how to address them. Talk about some other studies and research that are underway. We've heard about the need to better understand where PFAS are elsewhere than just in drinking water. And so again, we talked a little bit about biosolids. We are moving forward with additional studies, just got some additional monies to move forward on that, and monitoring, expanded monitoring of land application of biosolids in agricultural fields. Also, we'll be launching a study of soils around the state to better understand what we might anticipate finding in soil, because we are finding PFAS in soils, and we're not so sure how that is in context with other parts of the state. And we are not sure what the best method is to understand how much of the PFAS in those soils might actually leach down into groundwater. So that statewide soil survey is going to be huge. Also starting to look at plant uptake, working with USDA, FDA, and our Department of Agriculture and Rural Development, and Department of Health and Human Services, looking at what's being taken up in homegrown produce, even in some of the communities that have had higher impacts of PFAS contamination in the past. And lastly, we've deployed filters, like I mentioned earlier, in communities where we've seen impacts to private residential wells. And now our Department of Health and Human Services is going back to see, are those filters doing what we want them to do, especially since many of them were only certified to address PFAS contamination for a couple of compounds, and we're now concerned about several more at lower levels. And I should mention as well that we are trying to get a better handle on PFAS at the ecological studies from bioaccumulation risk for fish and wildlife. There's a study going on up in Clark's Marsh. We're looking at microorganisms and as well, and movement of the contamination throughout the food chain. It's not PFAS is not seeing behave like some of the other persistent toxics, so we want to get a better handle on that so we understand the overall risk in the ecosystem. And I am not the public health expert, and I've kind of lumped a number of different studies, but I think, from our previous presenters here, you can see great value in what our Department of Health and Human Services is doing, working with CDC, ATSDR, and others, trying to understand, especially in impacted communities in Michigan, where we know the exposures were higher because of really high concentrations in groundwater or even their public water supply, for example. So conducting exposure assessments, which can ultimately lead to some health studies, and doing some biomonitoring to understand what the state of Michigan population, what you might find in all of us, and as well as looking more specifically at firefighters who've been using those AFFF agents all along, and what has been their exposure over time, and trying to develop an investigation up in the Oscoda area for the former Wurtsmith Air Force Base that might help better understand what the exposures have been there over time. And I do want to acknowledge too that Michigan is not doing all of this alone. And in addition to coordinating all of our agencies and partners here in the state, we are working with others. And this is just one example where the governors and premiers in the Great Lakes region directed their states and provinces to coordinate on PFAS. And we have begun doing that in 3 areas that I think are really of interest here, because as Dr. Sunderland mentioned earlier, we really don't know a whole lot about air quality, air deposition. And so Michigan is actually leading a 22-state work group to better understand exactly that and where we go, and working with EPA and the other states. As well as we've got this issue with land application, not just of biosolids, but also concern over land application of other industrial byproducts. And so the states are coordinating on that, and fish and wildlife, as I mentioned before, how important that is to understand as we move forward, how PFAS are moving throughout our ecosystem. And I do have to acknowledge that there are some challenges or opportunities, if you will, as we go forward, is that, we've got the science is evolving very quickly. We all know that. We may end up with more standards that we need to contend with in drinking water and other media. And we've got those million private wells out there that we're going to have to address. Many of those will not be part of a formal MPART investigation. So we've got to educate those residents on what they can do. And the long-term infrastructure needs for drinking water, those are important, and I'm not so sure that we've got those totally figured out, and we're working on that as best we can with the resources available. And again, it's the cost, of trying to do all this, and it would be really helpful if we had some national leadership on this and some consistency amongst the states. I think it continues to confuse the public when states have different standards or criteria. And that's where I think hopefully moving forward that we get some more of that leadership from EPA, whether it's drinking water, biosolids, surface water, et cetera. So I will stop there and appreciate the time.
Well, thank you so much, Steve, and all the speakers for these excellent presentations and for getting us thinking about the many challenges surrounding PFAS in Michigan and elsewhere around the world. So we do have a lot of questions already in the Q&A box. So I think I'll start going through those, but audience, feel free to add more as you go along. If you cannot access the Q&A box for some reason, raise your hand and I'll see if I can let you speak. So,
when Jackie is going through the questions, I can use my host prerogative and ask a couple of questions. One directed to Dr. Sunderland and DeWitt. And I think you're all modeling PFAS exposure, and you talked about grouping them by classes or individual. Has anybody thought about modeling what is the actual exposure pattern? Obviously, there is blood levels, and grouping them in the concentration that they are up there. I think modeling it in an in vitro system and see the cumulative burden might give you a real life situation rather than looking at one. And for one thing, we don't know what all these PFAS are doing in terms of their action, how they are working. The question I have for Dr. Sliver is on the biosolids he brought up. And I always wonder, we all educate people to go for organic food, and biosolids are used in that, which could be a sink of ADC chemicals. And I think I would like you both to address that before Jackie groups all her questions.
All right, should I go first on biosolids? And I didn't catch all of the question, but the biosolids studies, and again, we've got a lot more work to do, but we know that PFAS, and in particular PFOS, is in the system, and you'll never get rid of it totally, but we can accomplish great reductions working with industry over time. But it's in personal care products and whatnot. So I think what's really critical is getting some criteria to where we know if you exceed that, and in particular PFOS is the one that our water resources folks and their team seem to be most concerned about right now, the most bioaccumulative, is finding out what that level is, what that screening criteria should be, so that we can find alternatives for those biosolids, or not biosolids, just sludges, until we're able to reduce the loads over time. I think they'll also tell you they've had great success, and once they've identified an industrial user and cleaned up that source, is that the impacts are not long term. But we've got this window here where we've got some biosolids we may have to deal with over time. I don't know if that fully addressed what you were looking for.
No, I was thinking a lot more than that. I was thinking a biosolid as a mixture or a sink of so many EDCs. How do they all interact with PFAS in terms of a cumulative exposure burden? I mean, are we doing justice by promoting organic food, which could be grown under biosolid conditions?
That's a much deeper question, and I do know that we do see value and the nutrient value in biosolids, and there is value as well as better understanding what some of the potential impacts are of what's in them, and that's the whole purpose what we're doing with PFAS right now.
And if I could just add to that, I think this idea of PFAS mixtures is a really important one. And I think the community is just starting to think about how to handle the toxicological impacts of mixtures and doing some lab tests. I think it's interesting to look at the regulatory approaches, and Steve outlined some of these, because a lot of them are assuming additivity. And so, teasing that out with some animal experiment data, doing, more sophisticated epi studies to think about the mixture problem, I think is on the horizon. And for the biosolids, in the data that I've seen, it seems like it's the biosolids that have an industrial sludge source that are the highest concentrations. So some of the biosolids have very low, even if they did receive, sort of a wastewater-ish impact. It's the ones that also have industrial inputs. So for example, in Maine, you've got an example of a pulp and paper sludge entering the biosolids and that actually having very high concentrations. I'm not sure if you're on mute and you're asking a follow-up.
No, I was just curious, is anybody modeling PFAS exposure in vitro system, like a real life exposure? You have levels that you have measured in blood. So if you really model that, or you put a mixture of PFAS in levels found in blood, what's the cumulative outcome, whether it's an immune response or whatever response you look at?
Yeah, and I think that it depends. So then that becomes an epidemiological biostatistics question as to how you handle those mixtures independently. I think to some extent that's been limited by the analytical chemistry. So the early studies, I think Jamie mentioned this, looked at just PFAS and PFOA. Now people are typically reporting 6. I showed you an example where we were using the full suite of targeted compounds to better understand exposure sources. And by the same token, you can use those characteristic exposure source mixtures and associate them with the health outcomes. But that hasn't been done widely yet. And it easily could, in my opinion, easily being caveated, of course, but it could be done as part of an epidemiological study.
And I think too, there are some challenges too, depending on where the source of the PFAS comes from. So if it's an aqueous film forming foam, we now know through non-targeted approaches that some of these formulations contain previously unidentified precursor compounds that can be metabolically transformed by organisms. So we did some work with Carrie McDonough, who's now at Stony Brook, who did her postdoc with Chris Higgins at the Colorado School of Mines. And she identified compounds in the serum of mice that were not in the AFFF formulation with which the mice were dosed. So that complicates looking at mixtures depending on the source because of potential for environmental and biological transformation products. So which ones do you look at?
Yeah, thanks for that discussion. And I know off the top of my head, some researchers such as Angie Slitt in Rhode Island and Chris Casotis at Wayne State are doing some in vitro talks where they model mixtures that were found in the environment. But even then it's, maybe 3 or 4 PFAS together, not 20 or 1,000 of them. So we have a lot of great questions now in the box. I'll try to go in order here. From Laureen Hackett wants to know, do you know of any research being done on health effects from inhalation exposure, lungs, or neurological effects from PFAS air emissions, such as those living near the emitting industries?
I do know of at least one researcher at North Carolina State University who's been looking at some inhalation effects, and he's looking specifically at immunological effects at the level of the lung. And he's found some interesting results associated with macrophage, which are some of the innate immune cells that wander around the lungs looking for pathogens that come in. He hopefully will be presenting those data soon. And his initial study was with the PFAS known as Gen X. But this is definitely an area of toxicology that's understudied.
Thank you. And we actually have at least 2 people have been wanting to ask Steve about the firefighting foam collection. Basically, what do you do once you have it? How is it disposed of? Is it incineration or something else?
Yeah, we made the decision last year when the program began to send it to a landfill after stabilization because the jury was still out on the effectiveness of incineration. And from what sort of incineration technique would be appropriate, because no one had really been studying that, to you didn't have a nationally validated stack test to understand what the emissions were. And so there was a lot of questions about it. It ended up going to a U.S. ecology facility in Idaho, in a very arid part of that state, to where the amount of leachate generated would be minimal. And so, again, helping to reduce the possibility of perpetuating that cycle.
Thank you. So we have a question now for Dr. DeWitt from Sun-Kuhn Park. He wants to know, do you have data or evidence that PFAS reduce efficacy of the influenza vaccine? And do you expect that it might influence COVID-19 vaccine efficacy? And also, is this the same in young or old people, or is it different?
So there's actually been a surprisingly few number of publications on the influenza response. I know of I think, at least 3 human studies that have looked at the influenza vaccine. And I believe that the findings are mixed. Sometimes it depends on the strain of the vaccine that's being administered. There have been, I think, only 1 or 2 animal studies looking at influenza responses. You know, mice don't get flu, so you have to give them a modified flu to determine if they develop those appropriate responses. And the results are mixed in terms of an influenza response. With respect to COVID, I actually just was invited this week to review a manuscript tracking the relationship between PFAS exposure and COVID infection. I can't say obviously by who or for what journal. But this is a question that has been brought up by many community members across different communities of PFAS exposure. They're wondering if their PFAS exposure will increase the risk of them getting infected, the severity of their infection if they are infected, and their body's ability to make vaccines once a vaccine is available. And then with respect to responses in young versus the old, if you look at some of the antibody data associated with vaccines, children have a decreased response at a lower serum concentration compared to adults. And I don't know about aged adults, but this is something that Dr. Sunderland also might be able to comment on due to her association with some of the folks who are studying immune and other health responses in some human populations. Sorry to put you on the spot.
Which part did you want me to comment on, Jamie? Just the COVID?
Oh, I didn't know if you had any new information that was shareable with respect to some immunotoxicological responses.
Yeah, I think it's an area of, you may have seen the latest if you have a manuscript in review. I think it's an active area of work. And so several of my colleagues are looking at this and looking for the association with, yeah, I don't really have much to add, sorry. I try.
This is a new area associating chemical exposure with COVID, since we're still learning about COVID.
I think my colleagues who work on this actively believe that there should be a strong association, and there's some language on, I forget which public health agency, about a potential association.
ATSDR raised the concern.
Yeah, raised the concern
about PFAS exposure and the risk of COVID.
Yeah, it's not a large leap in logic.
So now we have another question for Steve. This is also from Sung Kyun Park. He appreciates MPART's efforts to lower PFAS drinking water standards, and I second that. I think it's amazing, the standards the state has set, and hopefully the rest of the country will eventually follow suit. But his question is about alternative approaches for the auto industry for the electroplating surfactants, since now the PFOS standards are very, very low. You know, that industry may have to use, other things. So I guess the question is, do you think they'll start using other PFAS that are not regulated? Or is this something that MPART is going to be able to help regulate and clean up?
Well, I can provide a partial response to that, and that was one of the reasons for that fume suppressant study that we worked on with the EPA's Office of Research and Development, that identified some other compounds that we're not regulating right now. But again, I think Dr. Sunderland pointed out that, and we're not looking for all of the PFAS out there right now, and there may be some formulations out there that we're just not aware of yet. So I think, going forward, we're going to continue to work with industry and researchers to make sure that, what's out there, that we know the most about it as possible. Recognizing in some cases, we're not looking for some of these things. It may be a while. We're in Michigan, I think we are at 41 separate PFAS compounds, and others are looking for maybe a few more, but there's others out there, as we heard as well, thousands more.
Yeah, thanks. And we have another question about MPART from Chris Clark, one of our students here at the University of Michigan. He says that he has drank contaminated water his whole life living in this state, and he's a current MS student in EHS, and he wants to know, how does one become involved with MPART?
Great question. And I think one of the things you can do if you're interested, we have a citizens advisory work group that is made up of residents from impacted communities around the state. Right now, I think we have 29 people, and they volunteered to help assist MPART in a couple of things. One of them is to help us engage communities and empower communities that have been impacted by PFAS. You know, we think we do a pretty good job, but we can always do better. So that's one of the charges of the Citizens Advisory Work Group. The other one is, how do we better educate and inform the general public about PFAS and what the work of MPART is? And so this is a group that gets together the second Tuesday of every month, 6 to 7:30. They started doing virtual meetings before anybody else did because they're from all over the state. And I'm really encouraged by the leadership and interest of the folks that are on it. If he's interested, he can reach out to me. There's a tab, I think it's just called MPART, out on at that URL at the end of my presentation, www.michigan.gov/PFAS response, and it'll take you to Citizens Advisory Work Group. If you're interested in being a part of that, just submit the registration form. It's not an application, just register. All are welcome. And we love to welcome you and get your perspective and let you help us inform the rest of the state.
Thanks. That's great to know that it's a very engaged process. I want to jump ahead to a question from Sarah Caspar, because she had also raised her hand a while back. Sarah says she lives in the Pennsylvania Southeast area and had worked for US EPA Region 3, including responding to Mr. Tennant's request, the citizen that's featured in the Dark Waters movie. She says she's tried to get organizations she belongs to set up roundtables for information exchange, and this hasn't been very successful. No support from US EPA and other organizations. So the question is, whom can she contact? They have 3 sources of PFAS in the area: AFFF, fracking fluids, and industry. So do any of you know groups that are actually helpful in that region or nationally that could be helpful in this kind of situation?
Well, I'll just start that Pennsylvania, a couple of years ago, started its version of MPART. We did a briefing for them on how we structured, organized ourselves. And so I would check with the state to see if they still have that version going forward. They were doing some initial testing of drinking water supplies. And again, I don't know what's transpired lately, but that would be one place to start, because they may have some resources available to you directly.
There's also a national association of affected PFAS communities. I forget the precise name of this, but you could contact my colleague Laurel Scheider at Silent Spring Institute, who works with this group quite a bit. And I think the idea in forming that group is to have, a sounding board for different community concerns, and to learn what kind of resources and advocate together. So I would encourage you to reach out. Again, my colleague's name is Laurel Scheider at Silent Spring Institute.
I think there are also some people on this call who are part of community organizations at both the local and the national level who might be able to put Sarah in touch with some folks. I think that there are some really great community-led advocacy organizations that have done a wonderful job of getting people together to talk about how PFAS can be managed, addressed, evaluated in their states.
Thank you. Now we have a question from Brad Upham. He asks, what are the key technologies being considered for removing PFAS from drinking water?
Well, I can start out with I think the go-to technology is granular activated carbon. It's an absorption technology. That's what has been used, in a lot of different industries, even the wastewater side. But ion exchange resins and filtration like reverse osmosis. But again, the challenge with those technologies is you're really just moving PFAS from one medium to the next. And again, that doesn't break the cycle. And so we're working with researchers, and I know there's a lot of other folks out there working separately on destruction technologies. I know U of M is working on that, MSU is working on that, trying to come up with a method that's practical, economical, scalable to address something like a drinking water supply.
And the DoD funded CERDIP projects, if you go to their website, there's a lot of very innovative work looking at some of the compounds that aren't captured effectively by GAC filtration, and those are mainly the short chain ones, and trying to think about so if you think about the structure of a given PFAS, you have that perfluorinated chain, and the key is how do you, what they call it, unzip the perfluorinated chain, and so break that apart. Which is really a goal as opposed to just zorb it to something and remove it. So there's a lot of resources that you could read about on the CERDIP website, and it's a really nice portfolio of research by a lot of really great chemists and engineers.
Thanks. So now we have another question from Diane Wolke. She says the Ecology Center in Ann Arbor has found 100% of samples of synthetic turf that were tested for total fluorine revealed fluorine or PFAS in blades and backing. These plastic carpet surfaces are in daycare centers, schools, parks, and many different places. So what steps are being taken to regulate these types of products, if any? They're not recyclable and they're usually disposed of in landfills or near waterways.
So we, in my involvement in MPART, we have not been looking at that yet. And, but I mean, clearly that's, one potential exposure pathway. We know the city of Ann Arbor tested their leachate from their compost piles that where they were composting, I think, food packaging and whatnot. So it's out there in so many different products and in so many different media. And our big focus right now has been on the drinking water pathway, and then starting to look at prevention, like things like getting rid of the AFFF. It's something on the radar to look at these other exposure pathways. We just haven't gotten to that yet. But if you have information, please share it. Again, what we want information to help direct where we should be looking next, and what's going to be the most beneficial to protecting public health. And then we would look to researchers like Dr. Sunderland and Dr. DeWitt and say, okay, what do you think about this exposure pathway and its overall potential impact? So we always looking for more information.
And I don't want to take us down a more depressing route, but some of my colleagues have just had a paper accepted on PFAS use categories, and the number of categories of products and processes in which PFAS are used is enormous, much larger than I ever thought it was. And that really does mean that where we look for PFAS, we're likely going to find them. So if we increase where we look, we'll probably find them. I used to think that was a stupid thing to say, but I think that's the truth now, because they've been ingrained into so many products and processes that removing them is going to be a monumental challenge. And so we really need to consider not using them at all, or using them only under very specific guidelines with really good oversight. And I'm happy to share that paper. I think it's in its preprint form.
So now we have a question from Stuart Batterman. He says, thanks for the presentations. Another potential exposure pathway is vapor intrusion. While not likely to be a dominant exposure pathway, since levels are high in food and water, some estimates suggest a significant burden from vapor intrusion. So has this been estimated over locations with high levels in shallow groundwater?
So I've heard a number of people talking about this question recently, but I have yet to see good data on it, if that answers the question. And there is, the suggestion that you could have some exposure through this pathway. I think it would be not as potentially broad a population as these other pathways that we have been talking about, but certainly this is a gap that different groups are talking about right now.
And we have a quick comment going back to the mixture discussion a little while back, is just for those working in this field to consider interactions with non-PFAS toxicants, which I think is a point Vasantha had been getting at as well. for those studying in this field. Another question from Kathleen Michaels. What about PFAS use in production and disposal of materials like plastics? So again, another possible source here, plastic carpet manufacturers have admitted necessary use of PFAS in the plastic fiber extrusion, and it's been found in synthetic. Oh, actually this goes back to turf. So I guess does anyone have information about anything else in the plastics industry other than turf? I know a lot of these sources, as we're saying, are kind of new to all of us. Okay, so we have a specific question for Dr. Sunderland. It's wondering your perspective on measuring EOF. So I think that's the total fluorine rather than individual PFAS in epidemiological studies.
So EOF is extractable organofluorine, so it's a proxy for total fluorine. So as we're talking about, as Jamie was talking about, all these compounds that we couldn't necessarily detect, or they're precursors and AFFF that then transform. And they may have intermediate precursors before they degrade even more into the terminal PFAA that we care about. So this is a very complex problem with thousands and thousands of chemical structures. So one way to simplify this is to look at the total organofluorine in a given matrix. So you can look at that in water, you can look at that in biota, and you can look at it in human blood. And there have been a number of studies, and we're doing this actively right now as well, that do this. There was one that came out recently for Sweden. It's been done in Chinese populations. And what you see is quite interesting. So the fraction, typically you compare the EOF, so the total organofluorine that you're measuring, to how much you capture using the routine EPA method. So we call that targeted analysis. And the fraction really varies. It depends, again, on the exposure pathways. The amount of, we'll call it explained EOF that you see in human blood is greater than you see in the environment. And that would be expected because humans metabolize a lot of those precursors. But so for example, in the Faroe Islands, where we've looked at some pilot data, the people who consume whale meat have the lowest fraction of unexplained EOF, whereas the ones who don't, so presumably have a greater consumer products exposure pathway, have a lower fraction of exposure, or a lower fraction of explained EOF. And that implies that there's a lot of precursors, of course, that are coming through these other exposure pathways. So I think this is something that we could add to a lot of the epi studies. It does require a little bit more sample. So many of these studies, the limitation is the volume of blood that's available. And that's very precious in these longitudinal cohorts. But this is something that we're going to actively do in the ATSDR study that I'm part of. In Massachusetts, and we're also pursuing this in the Faroe Islands cohorts that are led by Paul Vehi and Philippe Grandjean.
Yeah, that's great news. And I'm a current EPID researcher, so I'll try to save as much blood as possible for future studies whenever I can in cohorts. We have another question from Doris Solarius, who's asking, does any waste management company offer secure storage for AFFF or other PFAS containing wastes until an effective destruction technology is available?
I'll take a stab at that. I'm not aware of anybody that's offering long-term, storage for this, whatever that means, because I think there's a question about how long it will take to come up with a destruction technology that passes muster, that's proven, and that can handle larger quantities rather than bench scale. And in some states like Michigan, you can run into potential conflicts with laws governing waste management. that, you know, as far as there are limitations on storage, depending on what the regulatory structure you're under. So I don't know anybody out there that's advertising long-term storage. The company we're working with has been storing material temporarily until it can be shipped out in larger loads out to Idaho.
So we're almost at the end of our time here. I think I'm going to give one more question. Participants can look in the Q&A box. There's a few comments that everyone might be interested in. And for the last remaining questions we can't answer, I'll see if I can get an answer emailed to you, to the people who asked them. So for the last one for today, have there been any conversations about culpability related to PFAS pollution? Natalie's curious about the products or companies that are polluting and how involved they are in remediation efforts.
Well, I will start very briefly that state of Michigan's Attorney General has filed suits against chemical manufacturers and the makers of the AFFF. And that's just now getting underway. And then from a more direct perspective, you know, MPART, EGLE, the state agencies are working with the users of these materials to have them address the contamination on their properties. And where they're unable, that's where we're stepping in with state funds and or federal dollars to address contamination. So, and I know other states have filed suit as well. Everybody remembers Minnesota's case that was settled, I think, a couple of years ago now against, I think, 3M. So, yeah, I would. That's all I know right now about it that I could actually really talk to.
I think there's lots of conversations. Whether or not anything will result in monetary damages being paid to those affected is still up in the air. I think there's lots of cases that are ongoing and lots of conversations.
I really want to thank our speakers today and M-LEEaD for sponsoring us, and for the audience and your engagement in this topic. If you're interested, you know, in all things PFAS, stay tuned for future events. The Department of Environmental Health Sciences here is going to try to virtually screen Dark Waters, which is a dramatization about the Mid-Ohio River Valley PFOA contamination, and have a discussion to go with it later in the academic year. We also have some M-LEEaD events on environmental statistics coming up, especially a week-long event at the end of October. I know Steve mentioned that the state of Michigan is having a big PFAS workshop, as well as the National Academies of Sciences. So just stay tuned for a lot of these events, and hopefully we'll all see each other virtually in the future and can work on issues regarding PFAS contamination and toxicity together. So thanks again.
Thank you all.
Yes, thank you so much.
Thank you, everyone.